Retinal ganglion cells, or RGCs, convey visual information from the eye to the brain via the optic nerve. But RGCs and the optic nerve are highly sensitive to injury. Diseases like glaucoma and optic nerve ischemia usually result in drastic RGC and optic nerve degeneration. With hundreds of thousands of Americans affected by neurodegenerative disorders every year, new treatment methods to not only slow, but also rescue neurodegeneration are desperately needed. In recent years, virus-mediated gene therapy has emerged as a promising treatment method. Specifically, AAVs, or adeno-associated viruses, have successfully been used to treat inherited genetic disorders in the retina and are highly efficient at infecting RGCs when injected into the eye intravitreally. Genes packaged into AAV vectors become episomes, or independent genetic elements, when introduced into RGCs and are overexpressed. With a strong promoter element like CAG, AAV vectors can drive gene expression in the retina. But here, a unique problem arises. AAV is not cell type specific. When AAVs are introduced into the retina, they infect all cell types, including RGCs, but also amacrine cells and glia cells and bipolar cells. If you are trying to rescue neurodegeneration in RGCs and the optic nerve, you should be targeting specifically RGCs. A highly efficient and reliable RGC-specific promoter for AAV that sustains gene expression does not yet exist, or at least it didn't. In 2020, the Haoliang Huang lab at Stanford published this paper in the Journal of Neuroscience. Its key finding? A highly efficient and reliable RGC-specific promoter with sustained gene expression over the course of multiple months. Mouse gamma-synuclein, or MSNCG.
Gamma-synuclein has been selected by a low-throughput comparison with other promoters as well as the universal CAG promoters.
There are three key ways this paper validates the effectiveness of the MSNCG promoter. Firstly, by comparing its expression of a fluorescent protein to gold standard RGC labels in both live mice and human cell cultures. Secondly, by truncating the promoter and demonstrating that smaller size, mini versions of the MSNCG promoter still drive strong gene expression. And finally, by demonstrating its effectiveness in rescuing neurodegeneration by driving the expression of a CRISPR/Cas9 gene knockout system.
We characterize by observing into the scanning laser fundus imaging as well as ex vivo flat mount retina. And then we immunostain the retina with RBPMS, which is RGC neural markers. Also, we use retrograde tracing with the dye to specifically label RGC from the superior colliculus. From the retina, whole mount and retina section, we selected gamma-synuclein is stronger and the most specific RGC promoters. It's about 90% co-overlap with the RBPMS, and it's over 95% with the dye. The reason we want to truncate the promoter is that the limitation of the AAV is 4.7 kilobytes between the ITR. And some of the genes are pretty big, and we require the promoter to be small enough to be able to drive it in the expression in our RGCs. So that's why we truncate the MSNCG promoter into different serial sizes, including 1 kb, 0.6 kb, and then finally we finalize with a 0.27 kb, which is still drive the GFP very potent in the retina, although the specific sense is a little bit not as specific as the full length. However, it's still the most specific one among others. It's still over 85% overlap with the RGCs. After optic nerve active injury, DDIT expression will be upregulated. So that's why we suspected that downregulation of the DDIT gene will have an effect of protecting the RGC from neurodegeneration. Also, somewhat knockout has been reported as the neuroprotection in the axons. So that's why we like to combine these two genes in the CRISPR-Cas9 knockdown system to be integrated into the small version of the 0.27 kb mouse MSNCG promoter, and we tested to and verify that these two genes combined can rescue the active optical crush model to protect both the RGC soma and the RGC axons by twofold from the wild type control. Then there is a significant rescue of both RGC soma and axons.
What's really important about this study is that it establishes a powerful and robust tool for RGC-specific gene therapy. This is a proof of concept study that establishes a baseline for new, innovative methods to induce regeneration and rescue sight. Every paper that the lab has published since this one relies on or uses the MSNCG promoter. This finding has immense potential to drive even more innovative findings in mice and one day in clinical trial.